Today we're going to talk about the experiments used to determine genetic code. The first experiment was done by Nirenberg and Matthaei in 1961, and they determined that a specific RNA sequence could code for a specific amino acid. And they first synthesized mRNAs that were homopolymers, which are RNA molecules with a single nucleotide. For example, they generated RNA molecules with all uracil nucleotides and only UUU codons, and they called this poly-U. And knowing that there were only 20 amino acids, they added poly-U, this cell-free extract, and 20 amino acids, with a different one being radioactive, into each tube. And then from there, they synthesized a new protein when they were incubated and processed. And then once that protein was synthesized, the protein was then filtered out. And if the protein was radioactive, then the labeled amino acid from the test tube must be in the protein, and therefore the codon that codes for that amino acid is such. And they ended up with UUU coding for phenylalanine. And they repeated this process with poly A, poly C, and poly G. And from poly A, they got lysine, from poly C, they got proline, and from poly G, it ended up being inconclusive. And the second experiment was done by Nirenberg and Leder in 1964. And they developed another technique that used ribosomes-bound tRNAs, because they found out that short mRNA sequences, even just one nucleotide, would bind to a ribosome. And the codon on that mRNA would base paired with the anticodon on the tRNA, and then that tRNA carries the amino acid specified by that codon. So for example, they used GUU mRNA, and it would bind to a ribosome, and then that tRNA with the anticodon would bind to the mRNA, and this tRNA carrying the corresponding amino acid, which would be valine. And then once they did that, they would filter out the unbound amino acids, and then the bound ones would bind to the radioactive paper, and then they would be able to see from there the specific amino acid. And they ended up with 61 identified codons, which are now in our codon chart that we use today.